In our last interaction, we have discussed about the various characteristics on the basis of which bacteria are classified, goals of classification, general methods used for classifying bacteria. We have also learned about how bacteria are classified on the basis of staining, size, shape and arrangement. Today, we will discuss about some of the other characteristics used for bacterial classification and major systems of bacterial classification. Flagella are the complex filamentous cytoplasmic structures protruding through the cell wall. These are unbranched, long, thread-like structures mostly composed of the protein flagellin, intricately embedded in the cell envelope. They are about 12 to 30 nanometers in diameter and 5 to 16 micrometers in length. They are responsible for bacterial motility. Motility plays an important role in survival and the ability of certain bacteria to cause diseases. On the basis of flagella, bacteria can be classified into various groups. A-trichous bacteria. A-trichous bacteria are non-motile bacteria. There are no flagella on the bacterial cell. For example, Diphtheria bacillus. Monotrichous bacteria. Monotrichous bacteria have a single polar flagellum. Example, Vibrio cholerae. Amphitrichous bacteria. Amphitrichous bacteria have one flagellum each on both sides. Example, Alcaligen faecalis. Lophotrichous bacteria. Lophotrichous bacteria possesses tufts of flagella at one or both sides. Example, Spirillum. Peritrichous bacteria. Peritrichous bacteria have numerous flagella all over the bacterial body. Example, Salmonella typhi. Bacteria can be classified nutritionally based on their energy requirements and on their ability to synthesize essential metabolites. Bacteria which derive energy from sunlight are called phototrophs. Those that obtain energy from chemical reactions are called chemotrophs. Bacteria that can synthesize all their organic compounds are called autotrophs. They are able to use atmospheric carbon dioxide and nitrogen. They are capable of independent existence in water and soil. They are of no medical importance. Some bacteria are unable to synthesize their own metabolites. They depend on pre-formed organic compounds. They are called heterotrophs. These bacteria are unable to grow with carbon dioxide as the sole source of carbon. Their nutritional requirements vary widely. Some may require only a single organic substance like glucose. Others may need a large number of different compounds like amino acids, nucleotides, lipids, carbohydrates and coenzymes. Saprophytic bacteria. Saprophytic bacteria obtain their food from dead organic decaying substances such as leaves, fruits, vegetables, meat, animal feces, leather, humus, etc. They secrete enzymes to digest the food and absorb it. The breakdown of carbohydrates is fermentation and of proteins is putrefaction. Fermentation produces alcohols, acetic and other organic acids by fermentation of carbohydrates. Putrefaction decomposes proteins into ammonia, methane, hydrogen sulfide, carbonic acids. The secreted enzymes break down the complex compounds into simpler soluble compounds which are easily absorbed. Example is Acetobacter. Parasitic bacteria. Parasitic bacteria obtain their food from the tissues of living organisms, the host. They may be harmless or may cause serious diseases. The disease producing bacteria are pathogenic, which cause various diseases in plants and animals. Examples are Bacillus anthracis, Bacillus tetani, Corynebacterium diphtheria, Mycobacterium tuberculosis, Streptococcus pneumoniae, Vibrio cholerae, etc. Symbiotic bacteria. Symbiotic bacteria live in close association with other organisms as symbionts. They are beneficial to the organisms. The common examples are the nitrogen fixing bacteria, example Azotobacter, Rhizobium, Clostridium, etc. Rhizobium species live inside the roots of leguminous plants and form bacterial nodules for fixation of nitrogen from the air. Bacteria vary in their requirement of temperature for growth. For each species, there is a temperature range and growth does not occur above the maximum or below the minimum of this range. The temperature at which growth occurs best is known as optimum temperature. In the case of most pathogenic bacteria, the optimum temperature is 37 degrees centigrades. Bacteria which grow best at temperatures of 25 to 40 degrees centigrades are called mesophilic. For example, Escherichia coli. Psychrophilic bacteria are those that grow best at temperatures below 20 degrees centigrades. They are soil and water saprophytes and may cause spoilage of refrigerated food. Thermophilic bacteria are those which grow best at high temperatures 55 to 80 degrees centigrades. They may cause spoilage of under-processed canned food. Some thermophiles, for example, Geobacillus stereothermophilus, form spores that are highly thermoresistant. Bacteria can be classified by their oxygen requirement. Aerobic bacteria. Aerobic bacteria are the bacteria which can grow only in the presence of normal concentration of oxygen. They require oxygen to support reproduction. Example, Campylobacter jejuni, Escherichia coli. Anaerobic bacteria. Anaerobic bacteria are the bacteria which can grow only in the absence of oxygen. They reproduce in the absence of oxygen. Example, Listeria, reduced oxygen levels, Clostridium perfringens, Clostridium botulinum. Facultative bacteria. Facultative bacteria can grow both in the presence or absence of oxygen. They can reproduce either in the presence or in the absence of oxygen. Example, Salmonella, Shigella, Staphylococcus aureus, Bacillus cereus. Microaerophilic bacteria. Microaerophilic bacteria can grow only in presence of minute levels of oxygen. Genotypic systems. Woos has developed a universal phylogenetic tree for all living organisms. This tree establishes a tripartite division of all living organisms, bacteria, archaea and eukarya. His work is based on a comparison of 16 as ribosomal RNA sequences. These sequences are highly conserved and undergo change at a slow, gradual and consistent rate. They are therefore useful for making comparisons among different living organisms. Ribosomal RNA, rRNA sequence analysis has emerged as a major method for classification. It has been used to establish a phylogenetic tree. In addition, it is now also used to rapidly diagnose the pathogen responsible for an infection, to help select appropriate therapy and to identify non-cultivable microorganisms. Molecular subtyping. Sometimes it is necessary to determine whether strains from the same species are the same or different. Pulsed field gel electrophoresis PFGE is the most frequently used molecular technique. Chromosomal DNA is digested with a restriction enzyme that makes relatively infrequent cuts in the DNA and as a result creates large DNA fragments. The DNA fragments from the different strains are then run on a gel and compared. Different classification systems have been given for bacteria. Three kingdom classification. Three kingdom classification system was proposed by a German zoologist Ernst Haeckel in 1886. This system of classification was based upon the morphology, tissue system, nutrition pattern and functioning of the different parts of the body of the living organisms. He divided the living world into three kingdoms such as Protista, Animalia and Plantae. All one-cell organisms were kept in the kingdom Protista. It included fungi, protozoans, algae and bacteria. Animalia includes all animals while Plantae includes all plants. The most common system of classification in use today is the five kingdom classification. R. H. Whittaker proposed the five kingdom classification in 1969. Under this system, organisms are divided into five major kingdoms: kingdom Monera, kingdom Protista, kingdom Mycota or kingdom Fungi, kingdom Metaphyta or kingdom Plantae and kingdom Metazoan or kingdom Animalia. Kingdom Monera. Kingdom Monera consists of primitive organisms. The organisms are very small and single cell. They consist of prokaryotes, which includes species like the bacteria, archaebacteria, cyanobacteria and mycoplasma. Kingdom Protista. Protista are single celled eukaryotes and are mainly aquatic. It includes diatoms, golden algae, euglena and protozoans like amoeba, paramecium, plasmodium, etc. They are mostly marine and photosynthetic. Kingdom Mycota. Kingdom Mycota or kingdom Fungi consists of a network of thread-like structures called as mycelium. The bodies consist of long thread-like structures which are called hyphae. These organisms are mostly saprophytes or parasites and also symbionts. This kingdom of fungi also includes lichens, mycorrhiza, etc. Kingdom Metaphyta. Kingdom Metaphyta or kingdom Plantae are eukaryotic multicellular plants. They contain chlorophyll pigment which helps them to prepare their own food by the process of photosynthesis. This kingdom includes all types of plants like herbs, shrubs, trees, flowering and non-flowering plants. Kingdom Metazoa. Kingdom Animalia or kingdom Metazoa are heterotrophic, multicellular organisms. They lack cell wall, This kingdom includes all types of animals. For example, lion, peacock, humans, etc. The three-domain system is a biological classification introduced by Carl Woese et al. In 1977 that divides cellular life forms into archaea, bacteria and eukaryotic domains. In particular, it emphasizes the separation of prokaryotes into two groups originally called eubacteria, now bacteria, and archaebacteria, now archaea. The complete classification of bacteria is given in Burgess manual. In this interaction, we have discussed about bacterial classification according to their flagella, nutritional requirements, temperature and oxygen requirements. We have also discussed about the major classification systems used for bacterial classification. For a more detailed understanding of bacterial classification, you may use the Burgess manual.